I welcome you to this biology paper 2 representation. Specifically, it is 2021 GCE question 2. So this question is coming from enzymes. And let me not forget, if you are new to this channel, kindly subscribe, okay? Yes, you are helping me, and also I'm helping you, okay? So the question is saying, figure 2 shows the stages involved in an enzyme catalyzed reaction inside cells of a living body. Okay, so now under characteristics of enzymes, like the one illustrated on this part here, we can say enzyme can catalyze both forward and backward reaction. Now, what it means here is that an enzyme can be used to catalyze the reaction, to break down the substrate into products and remain unchanged at the end of the reaction. Okay, so an enzyme can catalyze both forward and backward reaction. So an enzyme can be used to build up small molecules into large molecules. Okay, now you and me, we are familiar with metabolism. So metabolism is the sum total of all chemical reaction occurs inside our living body. Okay, apart from that one, I'm saying metabolism is the sum total of all chemical reactions that occur inside the living body. Apart from that one, we have two types of metabolism. First one being is anabolic or anabolism. Anabolism is a type of metabolism where small molecules are built up to form big molecules. So anabolism, ana means to build up. So small molecules are built up to form large molecules, like the one illustrated in this diagram here. I'm going to explain. Okay, so we have the first one, which is anabolism. Then lastly, we also have catabolism. Kata means to cut or to break down. So catabolism is a type of metabolism where large molecules are broken down to form simpler molecules or smaller molecules. Okay, now I forgot to define what an enzyme is. So an enzyme is just a biological catalyst. Okay, it is just a biological catalyst. Now, what is a catalyst? A catalyst is any substance which can speed up the rate of a chemical reaction. Okay, so let me try to label the parts and also the stages. So we have part I on this big shape of an enzyme. So part I is an enzyme. Okay, part I is an enzyme. So, as you can see, this part I has a biconcave shape here. So this part here, it is called the active site. Okay, this part here, it is called the active site, a place where substrates bind or a substrate bind. Then the other part of an enzyme is called the allosteric site. Then apart from that one, we also have G and H. So G and H, these are small molecules. These are small molecules. Now, specifically, there are substrates. So, G and H are substrate. Okay. So, now, as you can see on stage 2 here, so we can say on this part, stage 1, I is a reactant, which is an enzyme, and also G and H, these are reactants. Okay. So, on stage 1, these are reactants. Then, on stage 2, we have found that we can see that G and H are bind on an active site here, forming an enzyme substrate complex. So as for stage 2, it is the enzyme substrate complex. Then at the end of the reaction here, you can see that on the first stage here, G and H were separated. Now at the end of the reaction, we can say stage G3, these are products which are formed. So an enzyme Z remains the same at the end of the reaction. It doesn't change. Okay. So now as you can see, G and H at stage 3 are joined together. Now I was talking about metabolism. So we can say this type of metabolism here, it is anabolism, because G and H are small molecules and are built up to form a large molecule, which is this one here. Okay. So the first one is saying, which is question A, is saying from stage 1 in figure 2, identify the enzyme. So as for enzyme, I'm going to say I. Okay, I is an enzyme. I is an enzyme. And an enzyme is a biological catalyst. Okay, is a biological catalyst. Then as for question Roman numeral number 2, a substrate. Substrate we can say G and D, H or H and G. Then B, Roman numeral number 1 is saying which stage shows the enzyme substrate complex. So I'm going to say stage 2 because I was saying when a substrate binds on an active site of an enzyme, it is going to form an enzyme substrate complex. Let me say complex, yes. So the answer is going to be stage 2. Then as for Roman numeral number 2 is saying label the active site of the enzyme on figure 2. So I've already labeled it. So I'm going to say it is this one, the same one I've labeled it. So the active site is this part here. The part which is seen like a concave in shape, like this one. Or you can label it on stage 3, like this one. Okay. So let's move on to the next one. This is question C. Question C is saying, which is Roman numeral number 1, what type of metabolic reaction is catalyzed by the enzyme in figure 2? Now I was saying, G and H are small molecules, and at the end of the reaction, they are joined together. So which means on this part here, we can say anabolism, because anabolism is the type of metabolism where small molecules are built up to form large molecules. So I'm going to say anabolism or anabolic. Okay. Then apart from that one, we can say, which is our question, Roman numeral number 2 is saying, under question C, is saying, name a region in the human body where such a metabolic reaction in C, Roman numeral number 1 above can occur. So I'm going to say this one here can occur in the liver. Okay, so the answer here I'm going to say it can occur in the liver. Then as for Roman numeral number 3 is saying, state the general term used to refer to enzymes that work inside cells. So here they are talking about enzymes that work inside the living cells. So I'm going to say intracellular enzymes. So intracellular enzymes, they work inside the living cells. Okay. Then lastly, which is question D, is saying describe 2 industrial applications of enzymes. So I'm going to say enzymes are used in brewing, in the making of beer. So we can say it is used in brewing. Then the last one, we are going to say it is used in baking. Okay, so an enzyme which is used in baking, it is the yeast. Yeast act as an enzyme. Okay, so that was the last one. See you next time. Thank you.